Great, thanks, and thanks for inviting me here. This is great. It's my second time in Oxford. Last time was in 2015, so it's really nice to be back. So I'm going to talk about a couple of projects as one that we're wrapping up at the moment on centipedes. And so centipedes, they're a really old group of octopods. They're venomous, obviously. They date back over 400 million years. They all have this modified that doesn't work. Modified pair of front legs that have venom glands that produce venoms, lots of proteins and peptides that they use for predation, defense, even mating some species. Pretty cool. And a few years back, Ronald and I and a few others looked at the composition between the different venoms from different orders. So there are 5 excellent orders that you see there. And essentially, we found a huge diversity of toxins. So 93 phylogenetically distinct toxin families, so individual independent recruitments of proteins into the venom. And none of them were shared across all orders. So there's a lot of toxin gene family recruitment and losses. And so there's basically at this level here, their venoms are evolving in parallel. So they're homologous, but they're evolving, to say almost independent. Well, they would be independent, but without having necessarily homologous toxin families, which is really interesting because then it's a really cool system for studying evolution using comparative approach between those lineages with a homologous system. Now, the other thing that we found was that there's, as you can see here in this heat map, here you have the species, different orders. Down here, this is a heat map of the number of toxins in each toxin gene family that we found in venoms. And by and large, you can sort of see that at the bottom large group here have more stuff than the rest. And the reason why that is, we thought maybe could be due to the evolutionary origin of the centipede venom gland. So this is a diagram that Michel drew when he scooped my PhD thesis with his PhD thesis. And essentially, there's this idea that venom glands from centipedes have evolved from weaponized epidermal glands. So that's what you see in the red here. So this is a cross-section of the forcipule, which is the venom-bearing leg. And then you see this gradual folding in to have a discrete gland. And that's really cool. So you have this chitinous duct surrounded by weaponized epidermal glands. And so essentially, centipede venom glands are composite glands. So each one of those epidermal glands look like this. They're connected into this chitinous duct called a calyx through a pore, and that has a one-way valve. And then you have some canal cells and 2 secretory cells. So that means that the number of secretory cells in a venom gland is directly proportional to the number of pores. The number of pores depends on the length of this calyx. So in most centipedes, this is an MRI reconstruction of a forcipule from this theropodon longicornis. And so this is a duct, and this is where it becomes porous. And this is where all those secretory units attach. So they're really long, so large glands, but very few cells. And here you can see the pores from the inside, taking one of the calyces and cut it in half and imaged it by SEM. And then if you then compare that to what the scolopendrids have, the ones with the complex venom, they have really long calyces. And because of that length, they have, I think Michel actually counted these, and I think he found that they have about 1,000 times more than the simple glands. So 1,000 times more secretory cells, and they're lined up perpendicularly to this calyx. And so we proposed that this was probably the oh yeah, I forgot to mention that toxins are produced in different locations in the gland. So not one secretory unit produces all the toxins. So there's this relationship, probably the relationship between the number of secretory units and the complexity of the venom. So and that's this idea that the venom could be under morphological constraints because of limitations on protein production. So we thought that, okay, so if you have many toxins, they need to have lots of things to help them fold and be secreted, and that becomes exponentially more expensive to do. So there's a cap on how many you want to make. So that was an hypothesis. We published it. It was very good. That was a few years ago. And then I figured maybe it would be time to actually test that hypothesis. It's been the basis of a lot of my ideas and never actually tested it formally. So This is work from a master student, Sebastian. He specifically said he did not want to do any bioinformatics when he came into my office. This is exactly what he did. So he selected, I don't know if you can see these. Yeah, sorry, you can. So these colors here indicate the different orders. And then in black, those are the additional species. So we did some denser sampling. A lot of them are in scolopendrids with a complex glands, but spanning several sizes of the different species. Now, the really cool thing is that in scolopendromorphs, this one here, that includes the scolopendrids, but also Scolopocryptopidae and Plutoniumidae. I don't know how they ended up with the last name, but it's pretty cool. But they have evolved complex glands independently. So that's quite neat. And then all these other ones have simple glands and are kind of the same, except for these two geophyllomorphs, where Strugamia, for example, has normal ones. So you can see this is a micro CT reconstruction that's from below. And this itty bitty thing here is the venom gland, so tiny venom gland. This is Henia vesuviana. The venom glands, so that's the head, forcipules, and the venom glands are down here. And so they actually span several segments of the body. So they're huge relative to the body, but they're still very simple. So that's the tiny calyx there. So it's, you know, big glands, very few cells. So, so we sequenced transcriptomes, milk venom, and did proteomics on those to characterize which ones were in the venom. And then we made a list of all the sizes, because most of them, apart from the Henia, it's a really nice allometric relationship between the venom gland size and the body. And then decided we need to define complexity, because complexity, what is that? So because we're thinking about venom production or toxin production, we first went with toxin richness. So basically the number of unique proteins and peptides identified in the venom. And then split that into two. So either just unique ones. So basically every toxin has its own set of cofactors that it needs to in the secretion pathway. Or that if you're within, in this case, 70% sequence identity, then you share some of that. So the increase in this richness is slightly less if they're more similar. And then we also looked at, we decided to define it as toxin family richness. And then basically the rationale behind that is that if you have toxins that are evolution related, they maybe share the same folding machinery, right? So the cost of a toxin gene family expansion is not the same as another recruitment. And then finally, we looked at toxin diversity, where you also look at the distribution of the toxins between families, right? So a higher diversity means more even distribution of toxins between families, and then less diversity, you have, say, most of your toxins in one and very few in the others. And basically those incur different costs because of the associated folding machinery. And then finally, we also looked at the net number of toxin recruitment events because of this highly dynamic process of toxin gene family recruitment and loss. And so basically to see whether it could be a constraint on the actual evolutionary processes themselves. So long story short, we identified over 2,500 unique venom proteins and peptides in a whopping 141 putative toxin families, and that's a lot. I'm not going to go through all of them, but what we did then was in collaboration with a friend and a colleague of mine, Kjetil Boye at the Natural History Museum in Oslo, tested the correlation between complexity and venom gland size and type. So I'm not a statistician by any stretch of the imagination, so this is relied on him for this. But basically, we tested which models that were fitted to the data, which ones fitted the best, and then looked for significance. And basically what we found was that there's a really nice and tight correlation between gland complexity and whatever measure of venom complexity that we find. So in blue here, that's the simple glands, and red is the complex ones. You see here where we cluster, you see a much larger slope in the complex glands than the blue ones. And that probably has to do with the length of a complex gland also determines the number of secretory cells, right? So there's a correlation there. You see an even stronger pattern in the family richness, which also supports this idea that it's the more secretory cells you have, the less constraint you have is on completely new machinery for folding. And interestingly, in toxin diversity, we actually see a negative correlation with the simple glands, suggesting that when you go beyond, maybe if you have a large gland, then you're probably a large animal. Maybe there's this idea of larger animals might have a larger phyletic breadth and of prey, and then spiders and snakes, it's been shown to correlate with venom complexity in different measure, but still, so that kind of puts a cap. So then you're going to have to choose which of those toxin families that they prioritize. So that's nice when it supports an hypothesis that I've been talking about for years, not ever actually having tested it. And so, the reason why, again, it could be larger body size in these giant centipedes, scolopendrids are the biggest ones. They do eat the most things. So, that kind of makes sense. The other thing is that, this complete reorganization of the gland, you have all these perpendicular secretory units, kind of looks a bit like cone snail venom gland, right? So here, this is work from Sebastian. And where they found different regions of the venom gland produce different toxins secreted for different purposes, right? So it's super cool. And so we thought, well, we should test that in centipedes. So I had a PhD student, Vanessa Schendel. She did a really, really great job. So she looked at scolopendra morsitans, and then over a 3-year period, we collected stings series of defensive stings. So she held the centipede and squeezed it a bit hard, and then started secreting venom. And collected those sting series just to look for, you have displacement effects within the gland. And she also had 2 groups where she first did defensive milking, and then waited, and then did electrostimulation, like the normal way. And then another group where we did the opposite effect, just in case. And then did a bunch of analysis. So we did some MALDI and some in-gel digestion and top, bottom up and some activity. And basically, the sting series, there's no change. This is kind of, you don't really get the full picture here because most of the variation, as you'll see, is in here, but there was no variation between the sting series. In contrast, the electrostimulated and defensive venoms, they were different. So there are most of these, so they have similar components, or there's some components that are found in both, but there are also some that are not found in the defensive venom. And also some things that are found in defensive venom that is almost not found at all in the electrostimulated venom. So among the larger ones, there were some beta pore forming toxins, and also several of these putative neurotoxins that have been described before. And so that difference is actually reflected in the activity that we find. So there's no difference in the insecticidal potency against crickets, which kind of makes sense. If you want to secrete a defensive venom, then it shouldn't be any more insecticidal than electrostimulated venom. But in F11 cells, it was actually only the defensive venom that at this concentration caused activation. So that's really neat, I think. But it doesn't actually say anything about, well, could it be just a result of putting electricity on a gland and making it squirt out everything, as opposed to nice and gentle secretion by its own glands? So we tried to tried many years to try and make these little cricket armors of parafilm. The problem with centipedes is that they, as Michelle also showed, they don't just randomly sting their prey. They find the best place and then inject venom right there. And it's like it's a well thought through exercise, which sucks when you're trying to trick it to give you venom for a thing. So instead, we did this depletion experiment. So we did mass spec imaging. So we dissected a gland, and then you fix it in not formalin, but you put in a paraffin block, and then we sectioned the whole thing and imaged every slide. And then these are some spectra from the whole gland. And basically what we're looking for here is loss of peaks, not gain of peaks, because that means that they have spent it, right? So electrostimulation and full gland, you have about the same ratio of all the peaks, which is kind of nice. That tells you that you get most stuff. In the defensive secretion, especially one of these was completely lost. And in the predatory one, there were a couple more, which is quite interesting. 2 minutes. Yeah, oh shit. Okay, yeah. Okay, no, that's and then thinking about the constraints, we then asked, okay, so do they still have this functional segregation between the glands? And no, it doesn't. Turns out they don't. So these two were the ones that are associated with predation. One is in the front and one is in the back. And so basically it means that this control is at a really, really fine level, right? And so how that happens is that they're extremely highly innervated by neurons. So this is this yellow backfill. So you break off the tip and then you put this dye in and it migrates down the neurons. And so here you can see that this is just full of neurons. And Vanessa was able to trace this neuron that goes back into the subcephalic ganglion. So it's not like it's its own thinking leg, it's actually controlled. And then also looking for neuromuscular junctions and synapses, staying for synapsin and GABA. And what you can see basically is that along the duct here, there's lots and lots and lots of signal. Here are all the muscles, so there's lots of networks of neurons and muscles, and they have a really, this is a thing that's quite well controlled. And if you then go a little bit down in detail and look at it by transmission electron microscopy, so this is a micro CT, this is where the gland sits, this is a cross section of that. That's basically what we're looking at here. And this is, remember this diagram, like how the secretory unit looks like. Here, we're looking at here, this is the one-way valve that I was talking about. And you see these really nice, fine cuticular projections. And to those, you have attached these villiform structures. And this is where you have this really, really strong synapsin and GABA signal. Whereas up here is where you start having more muscle and also lots of synapses. So what we think is happening is that these nerve endings are enabling it to control it here by pulling these together, which basically expands here. And that creates negative pressure. So if you have muscles that squeeze here, you have less coming out because that is against the one-way valve. If it relaxes and you contract those muscles, venom comes out. So it's like a negative control thing. And that's basically how the ancestral epidermal glands control their secretion, but without the muscles that squeeze in as well. So, complex type 2 glands, really interesting evolutionary innovation to escape, in my opinion, or this is what I think at least, to escape physiological constraints from protein production, so you can have more complex venoms to do more things against wider range of, say, opponents. And then this has kind of formed the basis for another adaptation, which is then is a basis for enabling control over venom composition. So basically, an adaptation to something else has been accepted to this behavioral control, venom modulation. And it's just such a cool thing to be able to study everything from, you know, a gland down to gene evolution, the same system and their interaction. That's, I think, is one of the most attractive things about Venoms. So Sebastian did all the first bit, Vanessa did all the second bit, and then we have lots of collaborators at University of Oslo, University of Queensland. Natural History Museum in Ronald has been a huge help with lots of stuff. Greifswald with Carsten Miller, who did the TEM, all the micro CT was Andy Somke, and then thanks for funding, and thank you very much for not falling asleep, all of you, which is nice. Thank you.